Now I'll talk a bit about diffusion, which is another kind of image that we get, and it is really concentrated on the microscopic properties of the white matter. So you saw before that there was lots of different kinds of structural image that we can get, but diffusion is particular for looking at white matter. And so we can measure from that the directionality of different fibres within the white matter, which allows us to trace out what parts of the grey matter are connected to what other parts of the grey matter, looking at where the white matter tracks go. Or we can look at things related to the microstructure, things like axonal density or the myelination of the axons. It's loosely talked about as integrity. And that gives us a measure to have some information about what's happening in the microstructure. It doesn't give us all the information that we want, but it allows us to see differences which might be occurring due to, say, disease, neurodegeneration. So from diffusion MRI, we can get this information about the microstructure, we can get information about anatomical connectivity, that is about the wiring between different parts of the brain. But to do that, we need to get lots of different images and put them together in order to get information about the different directions. And this can take anywhere from 5 to 30 minutes of scanning time or even more in order to get diffusion information. What we get out of that is, as I said, information about either directionality or microstructure. And you can see the directionality information pictured here. You can see the red lines on the left-hand side give you an illustration of where the principal direction is of the fibre at each location. And at each location we actually have more information about just the direction. You can see a tensor represented here, which gives us information about what the direction is, but also about the diffusion in other directions, about some more information about the microstructure that we have. And on the right hand side you can see a colour-coded version of the same directional information. And so you'll see these kind of representations when you're looking at diffusion imaging and diffusion analysis. You'll also hear about quantitative terms representing the white matter. These are sort of surrogate measures of what's going on and represent things like mean diffusivity, or MD, or fractional anisotropy, FA. And these are very common terms. You don't need to understand what they mean at the moment. This will be unpacked much more in the main course. But these are terms which you may have already come across, or you may come across in the intervening time. From diffusion we can also trace from each individual point to another point in order to trace out the tracks. And you can see illustrations of the tracks here, either as a bundle, as you can see on the top left there, or as individual streamlines on the right-hand side, or as a probabilistic map of how one point is connected to other points in the brain, which is what we've got at the bottom there. And we can use this connectivity information to do other things. We can use it to actually do a form of segmentation. That is to say, take the thalamus and find subregions of the thalamus based on its connectivity. So here's an illustration where you can see the thalamus broken up into subregions, or parcellated on the left-hand side, with the colours corresponding to what area of the outer cortex they were predominantly connected to. And so you can see that gives us a lot of information about the substructures within the thalamus, which we can't see at all in the T1 weighted image. There's no information about those borders that we would be able to see from a structural MRI. So diffusion MRI is based on the measurement of water again, but it's actually the diffusion of water which really matters in this context, which you might have gathered from the name. What's crucial here is that the axons that are in a particular bundle all travel in the same direction. And that means that water finds it a lot easier to diffuse along the same direction as the axons rather than across. And that means that we have different diffusion in different directions, different amount of diffusion. And that's what we measure. We measure how far the water diffuses in a certain amount of time. And from that we get information about what is the direction of the axons and also how tightly they are packed, how much myelin is there. That also influences the amount of diffusion that we would see, predominantly in the directions which are perpendicular to the ones that the axons are going in. We can also get information in grey matter, but it's a lot more complicated, and that's really an area which is undergoing active research still. But white matter is an established technique, and we can look at what's happening in the white matter, and that's where most of diffusion analysis occurs. And so we get information about the microstructure. As I said, the density of the axons will influence the amount of diffusion, the myelination will also influence it, but we get it averaged over the size of a voxel. So it's sensitive to changes at a microscopic level, but only when they're consistent across this kind of size of the voxels that we're seeing, so across a couple of millimeters. So we look at bundles of axons, not individual axons. We're also only able to measure with one image how much diffusion there is in one direction. And so in order to build up a picture of how the water is diffusing in all different directions, we actually need to acquire lots of different images. And we need to synthesize those together in the analysis. How it works in order to find out how much diffusion there is in a direction is to use a thing called the gradient coils. So the gradient coils in a scanner allow us to create a magnetic field which changes along a certain direction. So here you can see the big blue arrow represents the direction where the magnetic field is changing. And what happens is that any water molecules which are moving in that direction, they change their phase. That's a property of what happens to them. You don't need to understand the details of that at the moment. But if they're moving along that direction, they change. If they move perpendicular to that direction, they don't. And so what we see is that we see a reduction in the signal for water molecules which are moving in that direction. They don't have to move very far. In fact, they don't move very far at all, because it's really the microscopic environment that we're looking at. But any motion along that is something that we can measure, and we measure quite sensitively small movements. And you can see in the image on the right-hand side, in the blue circle, it shows you an area where there's been a drop in signal. And you can see that, you know, by symmetry, it's quite different from what you would see on the other side. And that's because if you look at the directions, which are highlighted in the circle in the middle, then you can see that it's in those areas where we've actually got the water would be diffusing in the same direction as the axons. And so they find it easier to move in that direction, so we get more diffusion there, and we see a drop in the signal as a consequence. And we can change the direction that we're sensitive to just by changing the amount of current that we put into the gradient coils. And so now we've got a different direction, one which is going across, and we see a different area which is getting darker in this particular image. And so what we're doing in diffusion imaging is we're building up this information through lots and lots of acquisitions of these different images, and then looking at which regions we have reduction in signal. So that's basically what diffusion imaging is trying to look at. The limitations that we have are that it is not sensitive to the exact, or it's not measuring the exact thing that we want. So it's not measuring an axon size, it's not measuring an axon density, it's not measuring the amount of myelin which is present. It's sensitive to those things, and if those things change, then so does the signal intensity. But it doesn't give us a measurement for them. It also doesn't measure single fibres. We need a group of fibres together on the same spatial scale as our voxels, so a couple of millimetres. And when you've got a voxel which is that big, that means there might be several fibres going into it, and actually they might be crossing over each other, or they might be what's called kissing, which is touching together and then going apart. In either case, it's more difficult to try and figure out what's going on, because there's not a single direction of stuff which is happening within that voxel. So that's more difficult, but something that we can deal with in the analysis. We get quantitative local measurements of things like mean diffusivity or fractional anisotropy, but we don't get quantitative measurements out of connectivity. So when we're looking at tracks and we're looking at the connections between different parts of the brain, we don't get a number out which we can use to give us a sense of how many axons, for instance, were connecting these regions together. It's more difficult to do diffusion imaging in areas which are moving themselves. So the brainstem is pulsating as a consequence of the cardiac cycle. And that makes it more difficult to get good diffusion imaging in that area. It's possible, but you need to pay attention to it when you're doing acquisitions. Standard acquisition doesn't do quite so well. We can deal with bulk movement of the head. There are things built into the sequences to deal with that. But actually, when part of the brain is moving differently to other parts, that's more difficult. So the brainstem is really the most difficult region to image in this case. We're also a bit more restricted in diffusion imaging by the hardware of the scanner, so we need particularly good gradients in order to do diffusion imaging. It's very reliant on the gradients. And so certain types of scanners, particularly older ones which might be in hospitals, might not be able to do some of the more funky diffusion recent acquisitions that you might want to do. They are a bit more reliant on modern up-to-date hardware. We're also sensitive now to fast imaging artifacts, because as I said, we need to get lots of different images to capture the diffusion in different directions. That means we can't afford to wait for 3 minutes for each acquisition. We have to get each image in a few seconds. And we can do that, and there are methods for doing that, for getting images fast, but we pay a certain price for that. And that's that they're more sensitive to certain kinds of artifacts. And here are the two main ones which the diffusion image is sensitive to. One is distortion, and that's on the left-hand side, and you can see that that's where an area of the brain is displaced from where it should be. So it looks like it's being pushed in at the top on the left-hand side as opposed to the right-hand side, which is a non-distorted example. Or eddy currents, which are currents which are induced into the scanner when we switch our gradient fields on and off very quickly. And they actually distort the whole image. So they tend to what's called skew the image or shift it or scale it. And they have an overall impact on what the image looks like, as you can see in the right-hand examples. And that's on top of all of the other artifacts which we had in structural MRI. So there are a number of things that we need to deal with. Both the distortion and the eddy currents are always going to occur in any diffusion imaging that we have. And we have very good analysis techniques for trying to deal with those problems. In terms of analysis, the basic steps in the diffusion pipeline include correcting for the distortions, so that might be the first distortion I talked about, or the eddy currents, or motion correction, which is also done to take into account the fact that the head may have moved between different scans of the different directions. We fit things like the tensor to find out what the direction is and get some other information. We can then model fibres and use those to do tractography, where that is tracing the fibres from one part of the brain through another part of the brain. And within FSL, we're going to do that with probabilistic tractography, which is something that you'll cover in the main course. And again, we're going to feed that into statistics. We have to use statistics in order to account for the noise in the images and the biological variability. We're going to use the same method of statistics that we use in every other case. We're always going to use the GLM. We have the same framework, which is something you need to learn once, and then it applies to all the different types of neuroimaging analysis that you're going to do. Within diffusion, that might be looking at changes in some of these white matter microstructural surrogate measures, such as FA, and how they may be different, so like we had in our Asperger's case. Or it might be doing tractography, seeing how we can use that to do parcellation or segmentation of subparts of some of these brain regions. In terms of acquisitions, the best parameters that you would have for a diffusion acquisition are much more hardware dependent than other cases, because they depend a lot on the gradients and what your scanner is particularly capable of. So again, talk to the people who run your scanner. Another aspect of diffusion images is a thing called B-value, and that represents how strongly it is sensitive to the diffusion which is present. And that's something that you need to pick. It's very common to pick something in the region of 1,000 to 1,500, but that will very much depend on the kind of scanner you've got, the kind of study that you want to do. Also, the number of directions is important. If you want to do tractography, you need more directions. If you want to just measure microstructural changes, you need less directions. The numbers that we have are rough guidelines. Again, it will depend on the scanner that you're using and the kind of study that you want to do. You also need to get some non-diffusion weighted images, and these are called B equals 0. That means that you're just not sensitive to the diffusion. The B value is equal to 0. And you need quite a few of those in order to get a baseline from which to measure how much the signal has decreased due to diffusion. And we would recommend to get one every 8 or 10 images. We need to have an even set of directions in order to measure how the diffusion varies with the direction, and we recommend getting those across a full sphere. It's possible to get them across half a sphere because it actually doesn't matter whether you measure in one direction or 180 degrees to that in terms of the amount of diffusion that you'll see, but it also affects the artifacts that you see. So actually there are benefits in getting them on a full sphere. So when you ask that question, ask for a full sphere. We used to recommend getting a sequence which reduced the eddy currents at acquisition, but actually, and the analysis techniques have improved so much that actually now we think that it's better to get better SNR at acquisition by getting simpler sequences which have worse eddy currents and then correcting for the eddy currents later on. Again, there's not a right and a wrong answer to this question, it's a question of pros and cons. But we would say that at the moment, you're probably better off doing the simpler sequences with better SNR and then correcting for the eddy currents in the analysis. In order to deal with the distortions that you see in the images, we need to get another kind of acquisition. We either need to get an individual field map acquisition, which I talked about briefly in the structural MRI part, Or there's another type of thing that you can get, which is called a blip up, blip down, or a phase encode reversed sequence, and that's where you change a particular aspect of the acquisition of the diffusion images themselves. So you would get some of your B0 images with a different direction for the phase encode. And so that's quite a straightforward thing to do on the scanner, and that then allows you to correct for the distortions in a much better way for the diffusion images, and that uses a tool called top-up, which we'll cover in the main course. Isotropic voxels in diffusion imaging are even more important than they are in structural imaging, but basically we always want them. For automated analysis, we always want isotropic voxels, so make sure that you get isotropic voxels. And don't do upsampling on the scanner. This seems to be slightly more prevalent in diffusion imaging, is to click that upsampling button to get magical better resolution. It doesn't work. You don't get better resolution. You just get something which kind of looks a little bit better, but actually causes lots of problems in the quantitative analysis. So just don't do it. And there's an option to either get single shell or multi-shell kinds of diffusion. Either of those can give you good results. Multi-shell is becoming a little bit more common. There are some advantages to that. But again, lots of different studies use single shell or multi-shell. There's no right answer to that. And that brings me on to functional MRI now. So in functional MRI, we're actually looking at dynamic processes within the brain. So both structural and diffusion looked at the anatomy. Diffusion primarily at the white matter and how things were connected with the axons, structural with the overall gross kind of tissues. In functional, we actually see what's changing, what's happening when the actual neurons are processing information. We actually do that by measuring aspects of the blood response. So you'll see hemodynamic activity talked about, or the hemodynamic response. And that's because we're looking at the blood. And we have two different types, as mentioned at the beginning, task-based or resting state. And there's actually also another kind of change that we can do. We can look at intrinsic contrast with bold, or we can actually tag magnetically blood with a technique called ASL. We're actually going to talk about bold imaging today and for the majority of the course, but there are techniques that we have for doing ASL and for analysing that within FSL as well. Functional imaging, like diffusion imaging, is based on fast imaging sequences. And you can understand that. We need to see the dynamic processes of the brain going on, so we're not going to spend a couple of minutes getting an image. We're going to get images much quicker. So we get images every second or so, and we accumulate those over a period of 5 minutes up to an hour, say, in order to do a functional experiment. In terms of the analysis that we do, there's analysing the task fMRI, so that's when the participant is doing a particular task, seeing what areas of the brain respond when that task is being performed in a different way from when that task is not being performed. Or resting state, which is looking at functional connectivity. That is, what areas of the brain functionally seem to respond similarly in a spontaneous way. So they're looking at the spontaneous activity and how similar that is between different parts. And that's different from anatomical connectivity, because you don't have to have a direct wiring between two areas of the brain for them to be functionally connected. They can be connected indirectly through a third area. And as I said, ASL is another technique which we can do, which is able to do some of the same things, but is very good at doing quantitative studies of things like cerebral blood volume, cerebral blood flow, looking particularly at pathologies and other aspects of studies which are very difficult to do in the bold-related ones, which we're talking about for the other conditions. And one example of that is, say, drug studies, where they take a long time for the drugs to take effect. ASL really is the only option in that case. In terms of what happens in the brain, there are a number of different physiological processes which go on beyond the anatomy that we talked about at the beginning. So here's our neuron again, and what happens when it's active is that it's receiving inputs through its dendrites, and then it's actually sending out information through the axons electrically. So it's sending out little pulses of electrical activity, or spikes, which travel down the axon into the axon terminals, which then connect into the dendrites of other neurons which process that information and then may or may not send information out themselves. So there's that electrical activity that happens in the axons. There's also chemical activity which happens at the ends of the axon terminals and the connections to the dendrites. And that happens at a synapse, and there's a little cleft. So it's not actually an electrical conduction which happens between those two, but it's actually chemically based. There are neurotransmitters which are released from the axon terminal, they're taken up by the dendrite, and that is actually the process of communicating between these two different neurons. It's a chemically induced process. And so that actually takes a whole lot of other different things into account and requires other cells to support that activity, and also particularly the introduction of oxygen and glucose in order to give enough energy for all of these processes to happen. So there's a lot of metabolic demand going on when you've got activity. And in actual fact, a lot of it is associated with the postsynaptic changes. So here you can just see a rough breakdown of what's going on. But the details of this are not important. What's important is that you require the oxygen and the glucose to be delivered in order to support that, and that comes from the blood. And what's even more important is that blood has two different oxygenation states, oxygenated hemoglobin or deoxygenated hemoglobin, and they are magnetically different. And it's the fact that they're magnetically different which allows us to actually see activity with our MRI scanner. So the changes in their magnetic properties allow us to see how the water molecules near those blood cells are affected by changes in their magnetic environment. And we use that information in order to get all of our information about functional activity in the brain. So in MRI, we are really concentrating on what's happening with the blood response to neuronal activity, and doing it very indirectly by looking at how the changes in these hemoglobin molecules affect nearby water molecules. And this uses a thing called the T2 star effect. So I talked about T1 and T2 relaxation as two different properties. There's a modification of the T2 one called T2 star, which takes into account things like other external magnetic fields which are nearby. And here it's the magnetic fields of the blood which we're interested in, but this example is what happens if you've got some metal actually in the subject. So here's a case of a subject who had braces, and that's perfectly safe to be scanned with, but the problem is that that metal induces quite a lot of magnetic change nearby, and you can see that a lot of the bottom of this scan has just been lost. The signal's just gone, it's dark. And that's because there's a lot of magnetic field change in the voxels there. And what we need is a nice uniform magnetic field in order to get signals back. Once we disturb that, we lose a lot of signal. And so here we've disturbed it a lot because of the presence of the braces nearby. We've lost almost all of the signal. In the BOLD effect, the blood oxygenation level dependent effect, which is this thing which depends on the oxygenation of the blood, we have tiny changes in magnetic field, but enough to have a measurable effect. Nothing like this magnitude of effect, but enough for us to actually detect and measure the changes due to the neuronal activity. So the BOLD effect is the crucial underpinning of both the task and the resting state that we're doing. And here's an illustration of what it does. So we have our capillary bed that's fed by our arterioles, and that's extracting oxygen in order to process the normal demand of the neurons which are there. And it's the deoxygenated hemoglobin which creates distortions in the magnetic field. The oxygenated one behaves very similarly to all the surrounding tissues, and so that doesn't have much of an effect. The deoxygenated one is the one that counts. And so the more deoxygenated blood that we have around, the stronger the magnetic field changes that we have, which means that we reduce our MR signal, because any changes in this field from uniformity are bad. If we increase our neuronal activity, many things happen. One is that the body overcompensates for the amount of extra oxygen which is required and puts in a lot of extra flow and volume of blood into that area. And so the net effect is actually that the concentration of the deoxyhemoglobin goes down. So we are overcompensating for the amount of oxygen we need to extract, because that's a safe thing to do to make sure we don't starve the neurons of their oxygen. But that means actually that we have less deoxyhemoglobin around, we have smaller disturbance of the field nearby, and we get better signal, we get more MR signal back. So the consequence of all of this is that actually when we have activity, our MR signal increases. And so if you look at a plot of the MR signal that you would get versus activity, it would look something like this. When the stimulus starts, there might be a small initial dip, but that's very compensated for by this big overshoot and an increase in the measured signal due to the fact that there's more blood around and a decrease in the deoxy concentration. When the stimulus ends, then we do the opposite. it decreases, it actually undershoots, and then eventually goes back to the baseline state. And so that's the bold effect, and that's a very important thing to understand in order to know what's going on when we're looking at fMRI images. Here's an example of what is a fairly low quality fMRI these days. You can do a lot better than this, but it gives you an illustration of the fact that it's not a structural image. You won't see the same anatomical detail that you would in a structural image, and it's also subject to the same kind of artifacts that we saw in diffusion, particularly the distortion. It's not actually subject to eddy currents, which is a nice thing, but it's subject to a different kind of problem, which is signal loss. So you see areas here where we've got lots of signal loss going on, and in those areas they look a bit like a bite's just being taken out of the brain, and that's because in those areas we have a lot of change in our magnetic field and we lose a lot of signal, a bit like the braces example was before. What's causing it is the sinuses. It's the air in the sinuses and the air cells near your ears which cause these changes in field. So air behaves differently from all the other tissues which behave magnetically pretty much like water. And so these air pockets create changes in the field nearby and they get rid of a lot of signal. So you have a lot of problems getting good signal in inferior frontal and temporal areas when you're doing functional imaging. As I said, we get these images quite quickly, sort of 1 to 3 seconds each. They are optimized to give us as much signal as possible in response to the bulb change, in response to this change in oxygenation of the blood. And that's crucial for us because it's quite a sensitive signal. We need quite a small signal. We need as much sensitivity as we can get for it. We would often acquire hundreds of volumes over the course of 10 minutes or so. And we would also, in an experiment, get an anatomical image, a structural image, which has high resolution. You can see the structures. In order that we can map activations onto that, we can use that for alignment between different individuals. And it gives us an insight into where the activations are within our functional image, which is very hard to determine purely from the functional image itself. So even if you're not have an interest in particularly doing structural image analysis, you would still be requiring structural images both in functional experiments and also in diffusion experiments in order to support this kind of relationship between different individuals and also to understand the anatomy better for that individual. So the limitations that we have with functional MRI are that we don't measure directly the electrical activity of the neurons. We are not sensitive to when there is a signal going down an axon. We're not sensitive directly to the metabolic and the chemical activity which is going on. What we are sensitive to is the blood change. So bold fMRI is what we're doing, and it's sensitive only to the blood change. It's also qualitative. So a particular number that we get out for an intensity on its own, again, means nothing. It's only in relationship to how that changes over time, whether that gets bigger when a task is being performed or not, which gives us information. We're sensitive to fast imaging artifacts like we had in the diffusion before, and we're also sensitive to a couple of others. So I mentioned the distortion and also the signal loss. So here you can see the signal loss shown very clearly in this inferior frontal area, and that is the area which is very difficult to image. You need to pay particular attention to the acquisition of images if you're going to try and do functional experiments in this area. There's also other physiological changes which are more of a problem in functional imaging than others because they dynamically change over time. So things like the cardiac cycle, the respiratory cycle, change the intensities of our images, and they change them more in the inferior regions again. Again, a lot of things happen around the brainstem, particularly as association with the cardiac cycle. And so we need to do things in order to try and correct for those. And we normally correct for those in analysis because they're not something that we can get rid of very easily at the acquisition. Motion is also more of a problem in fMRI than it is in structural imaging. We're much more sensitive to small motions and the effects that those small motions have. And so a lot of the analysis effort goes into trying to get rid of the effects of motion. But that being said, you should do as much as you can to try and minimize the amount of motion during the acquisition. As I said, we need to optimize our sensitivity as much as possible, and that's by optimizing T2 star sensitivity. And if we want to get signals back in these inferior areas, we have to do special acquisition techniques. ASL is an alternative to doing bulb-based functional imaging, and it magnetically tags the blood instead of using this oxygenated, deoxygenated marker, but it has worse SNR. So typically bolder fMRI is the most common for most studies, but ASL has particular advantages in terms of quantitating things related to the blood flow and the blood volume, and doing experiments where there are very slow changes like drug infusion studies. When it comes to functional acquisition, there are again some tips that we have. So again, use the optimized sequences for your particular scanner and site. Talk to the people who operate that, because they're the ones who have the best knowledge of what's the best to use on that particular scanner. Get a field map. So the blip up, blip down is not an option for functional MRI like it was for diffusion, in terms of actually getting the raw data in that way, but you can get a blip up, blip down based field map, or you can get what's called a traditional field map or a dual echo field map. Whatever it is, just make sure you get a field map, because that will help you correct for the distortions in the analysis, do better registrations, get better power in your group statistics. It's quite important. In terms of signal loss, again, as I said, if you want to pay attention to what's going on in the inferior frontal and temporal areas, then you need to try and minimize the signal loss at acquisition. We cannot fix it in the analysis stage. So it's something that you need to sort out when you're acquiring the images. And there are various different options for doing that, such as changing the slice thicknesses or angling things. There are certain types of different shimming that you can do and so forth. You don't need to know about the details of that. Ask the person who's operating the scanner for you. They'll tell you what's available. Isotropic voxels are again important. Make sure you get them. Don't do upsampling, as we've said before. Again, that applies across all of the different types. If you're getting a small field of view, that is a cut down portion of the brain, then that's a perfectly acceptable thing to do in your functional study. If you're only particularly interested in one aspect of the brain, then you can get images quicker that way and you can get more data. But make sure that you get one single whole brain image of your subject using the functional sequence, the EPI sequence, which is used for functional MRI. So you just need to get one. It's only going to take a few seconds to acquire that single image, but make sure you get it because it helps again with registration later on. And there is a lot of interaction between the kinds of things that you need to do when setting up the acquisition, how you design your experiment, particularly your experimental stimulus if you're doing task fMRI, and the kind of analysis that you do. And that's true in general, but it's particularly true for task fMRI. So there's a lot of things which impact on each other. And so you need to understand all of those aspects when you're doing the study, when you're planning the study. And that's all I'm going to talk about now for structural, functional and diffusion imaging. Now I'll just briefly comment on a couple of complementary techniques which are available. So MRI is not the only way to get imaging information about the brain, whether it's functional information or whether it's structural, and there are various different other ways. Here's just an illustration of different kinds of ways and how they vary in terms of how spatially specific they are. So they're more spatially specific as you go down here, how good they are at picking up quick changes. So again, the quicker ones are on the left hand, the extreme end of the horizontal scale in this case, and how invasive they are. So fMRI in this case is not invasive at all, and neither is MEG or EEG, magneto or encephalography. Those techniques can be applied. They give you different kinds of information, but they're not invasive. Whereas PET is more invasive. You have to inject a tracer. It contains a radioactive isotope. And other techniques are, again, more invasive than that. And different techniques will measure different things. And that might be really useful. So EEG and MEG particularly measure the electrical responses, and they can measure the precise timings of what's happening in the neurons. When are those spikes happening? When are those neurons actually firing? Whereas what we're detecting with the blood response is much later than that, and it doesn't contain nearly the same kind of information about timing. We might also be interested in something which is more about glucose uptake or metabolism. And we can get other techniques which measure that more directly. And that might be particularly important in some cases for pathologies, for instance. So think about what you need to measure, what's most interesting. And it may be that a combination of different techniques is what you need. And that's not uncommon, is that you need to combine different things in order to get information about temporal aspects or information about other aspects, as well as the good spatial resolution which MRI is often able to give you. In terms of the different types of complementary techniques for structural, the computed tomography or CT based on X-rays is the main one that you would see. There's also histology for post-mortem imaging. Diffusion MRI, there's not really anything else in the non-invasive range, but there are other things that you can do post-mortem. And for functional MRI, there's actually a whole lot, or functional imaging, the alternatives to functional MRI, there's a whole lot of different things which measure these different aspects of the physiology which is going on in the brain. There are also complementary methods with respect to different types of analysis. So I talked about modelling the cortical surface and the foldings that we have there, and that's done with a package called FreeSurfer rather than FSL. We've tried to make our techniques complementary so that you can feed the analyses from one end to the other. Both are used, for instance, in the Human Connectome project pipelines. These two methods are very complementary, and we would recommend that you also learn something about FreeSurfer in order to do these kind of analyses, which are not just helpful for structural, but also for functional analyses as well. And then finally, I'll just talk very briefly about the first aspect of analysis with FSL. So this is brain extraction. It's the simplest thing to do, and it's just something that we'll talk about now because it will help us when we're looking at the first practical that we're going to do, which is an online one before you actually start the course. So brain extraction is a tool which is just trying to separate brain tissue from non-brain tissue. You can see the example here. We've got the brain tissue in grey, and we've got all the non-brain tissue coloured in that orange colour. And this tool simply tries to make a separation of those so that what we're left with is brain tissue, which is the thing that we're going to analyse. It's a very simple tool. It doesn't require a lot of different parameters to be set, and it's very robust. It works across a lot of different kinds of images. So not just T1 weighted images, but other kinds of images as well. It does help if you've got a little bit of FATSAT applied in your images to start with, as I mentioned before, because that suppresses some of the nearby tissues to the brain, which look like brain tissues but are not. What we're aiming for with brain extraction is not to model the fine detail of the folds. That's what FreeSurfer is for. And that's what you see in the bottom here. That's an extracted brain surface. We're not trying to get that. With brain extraction, we're simply trying to separate out the brain tissue and some of the surrounding fluid, the CSF, from the stuff which is outside of the brain and clearly outside. So the surface that we get is very smooth and very simple like this. How it works is that it starts with a sphere inside the sort of middle of the head and expands that out to try and find the edge of the brain. In some places, if it's already outside, it will actually contract in order to find the edge. And it does that in a way that maintains a smoothness of that outer surface. So that all it's trying to do is separate stuff which is inside, which is brain tissue, which can include some CSF, from stuff which is outside, which is not brain tissue. When you run it, as with any analysis tool, it's not going to be 100% perfect. You'll find that there are little things which might not be ideal. So it's very common to find parts of the image where it's left a bit of tissue behind, which might be non-brain tissue, or possibly it's taken away a bit of tissue, which is brain tissue. And whether that's important or not depends on what you're going to use this for. So we use brain extraction as one of the pre-processing steps for registration, for instance. And in that, it doesn't matter so much. But if you're using it in order to, as a pre-processing step for quantifying the amount of grey matter which is in your image, it matters a lot. Because in that situation, you really need to know whether these little bits of tissue might look like grey matter, and which, if they do, then they'll add extra volume which isn't really grey matter. And if you've taken away a little bit of brain tissue, then you'll be missing that from your quantification too. So in some situations it's very important, but for registration, which is the one that we'll use at first in the actual course, it doesn't matter so much whether you've got some slight imperfections. And we'll talk a little bit more about this in the online practical. And that's it for this talk. I hope you've learned something through this process. Again, if you're new to the field, you won't understand absolutely everything that there is from this, but make sure that you've got to grips with the fundamentals. Rewatch it if you're not sure about some of the things. If you have been in the field for a while, I hope you've learned some of the extra things. There'll be a lot more information about this in the FSL course and a lot more opportunities to ask questions during the lectures and during all of the practical sessions. So if you've not sure about some of the things, then ask us during the main course. And so that's it for now, but make sure that you do look at the online practical before you get to the course as well. Thank you.